Greetings my dear students, I am Dr. Monica working as Associate Professor in NIET Pharmacy Institute, and that has been affiliated to Dr. APJ Abdul Kalam University, and the subject I am going to take is your Pharmaceutical Microbiology having code BP 303T. Now, in this lecture I will move on with the clean area classification. In the last lecture I have covered up, I have started with the unit number 4, and unit number 4 what we did, that is aseptic layout we have covered based on your classification, clean area classification. I covered up the aseptic layouts. Even I told you what actually clean area is, how we can classify our clean areas based on various guidelines, we can classify into various categories. So, based on clean area classification, various layouts being designed for your parenteral sections, for your microbiome labs. So, based on that is clean area classification, that aseptic layouts we have discussed in the previous lecture. And now in this lecture what we are going to cover, we will cover this clean area classification in detail, as well as along with that we will cover up the laminar airflow unit, that units which are the units which provide an aseptic area, which are being used for to provide an environment which must be free from microorganism, as well as we will try to focus on some of the sources which are there, which are responsible for the microbial contamination within the area. So, in this and also I will like to focus on the preventive measures which we can take, preventive precautions which we can take, so that we can prevent such type of contamination, we can avoid the sources of contamination within the area. So, actually the prevention is required in case of pharmaceutical products, because not only pharmaceutical, we have to mainly focus on the parenteral pharmaceutical products which we need to have, that those products must be sterile, that must be free from microorganisms. So, that is why we actually require those conditions, those that precautions, so that no contamination can take place within such products which are going to be delivered directly to the systemic circulation, which are being injected within the human beings, which have to be directly reach the systemic circulation. That products can't be contaminated with there should be no microorganism within those products. So, that is why we always take extra precautions when we are dealing with the parenteral products, when we are dealing with the ophthalmic products which have to be directly injected or directly put into the systemic circulation. Whereas, in case of oral preparations, or when the preparations has to be given through the mouth, in that case there we can prepare them into the normal labs. But when we are dealing with the parenteral section or parenteral products, we have to take extra precautions so that there should be no contamination within those products. So, now how those clean area has been classified based on various guidelines. So, that we will discuss. Various guidelines have given different type of grades, or they have an ISO also has given the clean area classification, but the production area is normally whatever production area is where we are going to actually manufacture our parenteral products. In that production area, or if we say about microbiological labs, those labs are always divided into 4 grades. Now, what are those 4 grades? Depending upon the limits. In my previous lecture also I told that depending upon the limits, which limits? That are the limits for dust particles, that are the limits for microbial contamination. Depending upon the limits of contamination, our regulatory bodies have given various grades where they have specified, they have classified the clean area into various categories, but which actually depends upon the limits of contamination, and limits of contamination depends upon the particles. The limit of contamination depends upon the microbes, that is how much microbes are there within some area, how many dust particles are there within any of the area. Depending upon the limit of dust particles, depending upon the limit of microbes they have, regulatory bodies have given various specification, various classification. But the classification we generally use in case of pharmaceutical industry, or the classification which we mainly use in pharmacy, is this one, that is grade A, grade B, grade C, grade D. We also call it as class 100, class 1000, class 10,000, class that is 1 lakh. So, these class 100 we also use the classification criteria like class 100, class 1000, class 10,000, class 1 lakh, depending upon the particles, dust particle, how many dust particles are there within that area, how many microbes are there within that area. Depending upon those limits we can classify, like grade A may also be known as aseptic area. Grade A is always known as aseptic area, and that also been defined as class 100. Grade B is having also called as what we call as gray, we can use the word white area for grade B. This is aseptic area, this is that is white area, this is gray area, this is black area. Means aseptic, that is white, black, gray and black. So, this zone is highly contaminated, that is why we call it as black area, and also we give the term as class 10,000, we use the term class 1 lakh, means it is having maximum number of dust particle, it will be having maximum number of microorganisms will be there in this zone. So, that is why it has been put under black area. But when we move from this black area towards gray area, that will contain less number of dust particles, that will contain less number of microorganisms. So, when we move from grade B towards grade A is the aseptic area, that will be having no microorganisms, that will be having minimum number of that is dust particles. We will not say that there will be 0 dust particles, but minimum number of dust particles at that limit have been specified with the help by the regulatory bodies. So, depending upon the limits of particles and limits of microbes, we classify them as grade A, grade B, grade C and grade D, that are also known as aseptic area, that is white area, gray area and black area, and also we can say them as class 100, class 1000, class 10,000 and class 1 lakh. So, in this way we can categorize, in pharmacy we use these 4 grades and we divide our microbiome lab into 4 grades. If whenever you will get a chance to visit the microbiome lab, in the microbiome lab you will see that whole lab will be divided into 4 zones or 4 grades, from starting from grade D till grade A. At grade A is the aseptic area, that aseptic area is required for the preparation, for the packaging part, for the that is sterilized products can be prepared, sterilized formulations can be prepared only in aseptic area, the area which will be free from microorganism, the area which will be free from that is particle contamination, the area which will be free from dust particles, that will have what we use the word HEPA. HEPA means highly that is high efficiency particulate air, means these will be having a filters, means the filters which will remove the dust particles, the filters which will remove the that is what we call as microorganism even from that aseptic area. So, HEPA filters are always are there within grade A. So, such type of filters are being added to those areas. Even we use UV lights, UV lights are always placed within that area, so that they make the environment clean, they make the environment free from microorganism, they make the whole area aseptic, free from microorganisms, free from dust particles. So, various equipments, various techniques we can use to make that area aseptic, to make that area free from microorganisms. So, aseptic area is the area which is being used for the preparation of parenteral products which we need to prepare, that must not get contaminated with any of the microbes or dust particles. So, aseptic area is the area which is free from microorganism, grade A. But according to if you go through this table, on the basis of this table mainly is based on maximum number of dust particles, maximum number of particles which can be there within a specified area, which are according to the regulatory bodies, which are being allowed. Out of, if the particle number increases, then that area also change its classification. If we can see according to the dust particle classification, if any of the dust particle is having a size, dust particle having a size 0.5 micrometer, if any of the particle is having, dust particle is having 0.5 micrometer size, that you can see in grade D it is, that is 3520000, means in lakhs of particles can be there within grade D of 0.5 micrometer size. But as we move from grade D towards grade C, and from grade C towards grade B, and from grade B towards grade A, the number of particle size, number of particles go on decreasing, means A as grade A is the aseptic area, the number of particles which are having 0.5 micrometer size, that are only 3520, whereas that was there in grade D, there were in lakhs. And if we see the dust particles which will be having size 5 micrometer, those particles are just 20. According to the regulatory bodies, they have given these guidelines. So, only 20 particles of 5 micrometer size. And if we go for that is what we mean by at rest and what we mean by in operation. At rest means when there is no working going on, we are not doing any of the work. When at that moment, if we analyze this that is particles that of 0.5 micrometer size and 5 micrometer size, so in that case this is the limits. And if the area is under operation, when we are doing work, there are a lot of personals working over in that area, operation is going on, equipments are working, in that case limits are little bit different. So, for grade when we move from grade D to grade A. So, you have to mention for both at rest as well as in operation the limits which has been given by the regulatory bodies, that how many dust particles are allowed of 0.5 micrometer size or 5 micrometer size. But more than 5 if the particle size or if the dust particles are having size more than 5 micrometer, then there should be no particle of the dust particles which have the size greater than 5 micrometer. Those particles are not at all allowed within that grade A. Aseptic area must not contain any of the particles which is having size more than 5 micrometer. So, based on maximum particle per meter cube, this classification has been given. These are the limits which you need to understand, you need to note it down, that limits which have been given by the regulatory bodies for grade D, grade C, grade B and grade A. After that, this table is actually based on microbiological monitoring. That means it is actually based on microbes. How many microbes can be there within a grade D area or within a grade C area, within a grade B area or grade A area. How many that is microbes are being allowed. So, in that case, if you see, this is the main specifications, that limits which are there. Suppose we are talking about grade D. In grade D, we can have only 200 CFU. Now, what we mean by CFU? That is colony forming unit. When we are that is observing, or when we are talking about the microbes, as microbes can't be seen with the naked eye, we can't see them as within the lab, or we can't see them. In that case, what we can do, we can that is incubate them, we can cultivate the microbes, whatever microbes are present within the environment. What we need to do? we have to make the colonies of those microorganisms. Now, how we can go for that? We have to go for different techniques. Like in this technique, there are air sampling techniques, there are settle plate methods, there are contact plate method, there are glove printing methods. There are various methods by which we can collect the samples. Now, that sample may be a air sample, that sample may be a surface sample, ki how much microorganisms are there within on the surface, how many microorganisms are there within the air. So, we have to collect those samples to analyze ki how many microbes are there within the air. For that, we need to collect the sample. So, various techniques can be followed, like settle plate, contact plate or glove print. So, what even cotton swab methods are there which we can use for collecting the samples, surface samples, for collecting the air samples, for collecting any of the samples which is of within the lab. Those samples are being collected, and after collecting those samples, we have to inoculate those samples within the media. In that media, what will happen? That media will contain nutrient media, and that nutrient media will provide the environment, that nutrient media will provide the nutrients in which those specified microorganisms, the specified samples which we have taken from the surface, the samples we have taken from the air, those samples we inoculate within the Petri plates which contains the media. That media will be made up of nutrients that will provide nutrients for growth of those microorganisms, and when those nutrients level we will give, and after that we inoculate. After inoculating our samples, whether it is air sample, surface sample or any of the sample in which we want to that is monitor the microbes, those samples are being inoculated into the nutrient media, and after inoculation we have to incubate those samples into the incubator. And what will happen? After 7 to 14 days, that samples containing microorganisms will form colonies. Colonies will be formed as microorganisms will grow, and colonies will be formed, and those colony forming units are being counted within the Petri plates. How many colony forming units are being there within the Petri plate? So, whatever the samples have been collected from 1 metric cube area, those samples are being inoculated in the media. After inoculation, we have to incubate, and after incubation we will see colonies will form, and those colonies will be counted with the help of colony counters, and those specifications have been given here. If colony which we see after 7 to 14 days within the Petri plate, if the number of colonies is less than 1, means that area will be grade A. If the colonies will be more than 200, then the area will be grade D area. So, if no microorganism less than 1 means no microorganism, then that area will be grade A, and if number of microorganisms will be more than 200, that area will come under grade D, that will be highly contaminated. If it is there, the colonies which we see, which are that colonies are that is more than 100, that more than or but less than 200, then that area is grade C, and if colonies are 10, that is more less than 100 but more than 10, then in that case the area is grade B, and if there is no contamination, the number of microorganisms are less than 1, then there is no that means that is aseptic area, that is free from microorganism. Whatever techniques we are using, we can see that number of microorganisms must be less than 1 in aseptic area, and in this case you can see there may be 5, in this case there may be 50, and in this case there may be 100 to 50. So, this is the way the methods, the techniques may be different for collecting the samples, that is for forming the colony forming units, but the limits, as you can say, limits for grade A will remain the same. There should be no contamination, the number of microorganisms must be less than 1. So, this is what we call as clean area classification. This classification has also been given by ISO nowadays. Now, ISO also have given this classification based on number of particles having size greater than 0 micrometer, greater than 0.2, greater than 2 micrometers, depending upon the size of particles they have given different classifications, that ISO 1, ISO 2, ISO 3 to ISO 9. 1 to 9 they have classified, that is depending upon the, as you can see in this case maximum contamination, in this case minimum contamination. When we move from this towards this, the contamination decreases, the number of dust particles decreases. So this is the classification depending upon the particle size, that how much of this particle size how much particles are allowed, 0.5 micrometer size how much particles are allowed, and 0.3 micrometer size how much particles are allowed. So limits have been given either by various pharmacopoeias or by ISOs. Depending upon the standards, we can vary the classification of our clean areas. So after that comes the laminar airflow units. We have to also discuss about this thing, that is what we call as laminar airflow. Laminar airflow is actually a unidirectional flow. Why we need a unidirectional movement or unidirectional flow? So that we must be able to remove microorganism, we must be able to remove that is dust particles from our area. So this is a special type of a unit which is being placed within the grade A. Grade A, always in grade A, which is also known as aseptic area, this is a special type of a cabinet having HEPA filters. HEPA filters are always there within that unit, and that HEPA filters are there to remove microorganisms, to remove air dust particles. So these HEPA filters are always there, either they are placed vertically or they are placed horizontally. They can flow the air will move from outside, it will go towards the HEPA filters, and then it will move towards the bench on which we need to work, we need to prepare our preparations. So in this case the flow may differ, but whatever air will come into this unit, that air will be has to pass through the HEPA filters, and when it will be passed through the HEPA filters, high efficient particulate air systems, highly efficient filters, when that air will pass on, that air will be filtered out, they will remove all the dust particle, it will remove all the microorganisms from the air, and then it will flow in the cabinet in which we want to work. That cabinet also contains UV lights. UV lights are also always attached to within this cabinet, so that this whole area will become microorganism free, that whole area will become free from dust particles. So that laminar airflow unit, laminar flow means unidirectional flow, we can't have that is turbulent flow within that area. If there will be turbulent flow, we won't be able to get the effective, we will not able to that we will not be able to make that area clean from microorganisms or clean from dust particle. Turbulent flow can take place from any direction. If the air is moving from any direction, means we won't be able to that filter it out, we won't be able to that is move, get a free area free from air free from microorganisms. So the air must move in a unidirectional way, it must not be turbulent in nature. So there are different types of that is directions of air. If HEPA filters are attached here, then we get air in this way. If this is the cabinet and here we are sitting and we have to work within this cabinet, in this cabinet if the air is flowing in this way from this direction towards this direction, means this is known as your horizontal direction, means air is moving horizontally in one direction, air is moving. So this is known as your horizontal. We also categorize this equipment as horizontal laminar airflow, and if the air pass from the top and HEPA filters are attached here and air is moving like this, means this flow is vertical. This is you are sitting here, this is a bench on which you need to work and air is flowing from top towards bottom, then this is called as vertical flow, but the movement of air remains unidirectional. The laminar airflow can be categorized based on the movement of air, whether it is horizontal laminar airflow or vertical laminar airflow. This is vertical one and this is horizontal type. If HEPA filters are attached here and air is moving, filtered air is moving from this side towards this side horizontally, then they are known as horizontal type. If air is moving from top towards bottom, vertical, so this type of units, this type of laminar air units are known as that is vertical type. So, flow, if we talk about flow, the flow is always unidirectional, means in this case as you can see only one direction is being followed. This is known as unidirection, either vertical or horizontal, and this what we call as non-unidirectional. If there is no direction of air, non-unidirectional, this is the diagram which is showing there are HEPA filters are there are air conditioning units, there are outlets for the air, but the air is moving, air is having no direction, that flow is known as non-unidirectional. We can also have unidirectional and non-unidirectional combined airflow system, in which there will be non-unidirectional flow as well as the unidirectional flow within the cabinet. So, we can categorize our system based on that also, that is whether they are unidirectional, non-unidirectional or combined airflow systems. So, this is laminar airflow. After that we have to cover the that is sources of contamination. Now, what are the main sources of contamination within any of the area? As we have discussed about clean area classification, but how our area gets contaminated with microorganism, how our area gets contaminated with dust particles. Now, what may be the sources of contamination? When we talk about our pharmaceutical products, when we are talking about our any of the pharmaceutical products or we can also talk about the lab area also get contaminated. We need to know about the sources of contamination based on whether we want to judge, whether we want to evaluate the sources of contamination within any of the lab, within any of the that is manufacturing unit, the sources of contamination within the pharmaceutical products, that may differ. What are the sources of contamination within any of the area, what is the sources of contamination within any of the pharmaceutical products. So, sources of contamination actually generates from facilities, the area, the equipments which are we are using, the raw materials which we are using within that area, the personas which are there within that particular area, that all are the sources of contamination. The contamination may take place from the environment, from the windows, from the doors, from the water samples which we are using. The contamination may take place from the personas who are working within that area. The contamination may take place from the process which we are using for manufacturing. The contamination may take place from the utilities present within that area. So, all are the sources of contamination, whatever raw materials we are going to use, whoever the personas are working within that area, whatever the equipments are there within that area, whatever the airflow units are there, whatever the light sources, the buildings in which we are going to work, that all are sources of contamination. They can contaminate our products, they can contaminate our environment. So, we have to take precautions while working in that areas. We have to take care what raw materials we are going to use. Those raw materials must be sterile. Whatever raw materials we are going to use, we have to sterilize those materials. Whatever that is equipments we are going to use, we have to sterilize those equipments, because they are also the sources of contamination. Whatever glasswares we are going to use, those glasswares must be sterilized by any of the techniques. Technique may be hot air oven, dry air, the technique may be autoclaving, but we have to take preventive measures. We have to take source of contamination, maybe your personas who are working, the students, the lab technicians, even the faculty members, whomsoever is working there, that may be the source of contamination. So, we have to take extra precautions while we are working within the aseptic areas. We need to take care for the personal hygiene. The buildings in which we have made that lab or the environment, that may be also a source of contamination. We need to sterilize our environment, we need to sterilize our air with the help of UV lights we can add. We can go for any of the sterilization procedures like disinfectants, we can use disinfectant, we can take preventive measures to so that we can control the sources. First we need to know the sources, and after that we need to know the preventive measures which we can take so that we can avoid such contamination within that area. So, sources of contaminations can be personas, that can be production processes, that can be environments, that can be material equipments, that can be airlock systems, whatever is there within that area, that all are the sources of contamination that can contaminate our area, that can contaminate our pharmaceutical products. So, we have to control all these things, we have to take preventive measures, we have to take extra precautions while we are working within that any of the area. So, preventive area, the preventive measures, as I told, if equipment are there you have to sterilize, if glasswares you are using you have to sterilize, airlock systems you are using you have to take extra precautions to see these conditions. Building, if there are light sources, the light sources must be fitted with the ceilings, there should be no entrapment within the light sources, there should be no chances of entrapment of dust particles, entrapment of microbes within that systems. So, all doors, windows should be fit flush with the walls, whatever windows you are using, whatever doors you are using, whatever pipes are there, gas cylinders are there, you have to take extra precautions while installing those. So, you have to go for the personal hygiene too, you have to use the gloves, you have to use the mask, you have to use the lab coats, that all must be sterile, and you have to take extra precautions while working within that area. Personal hygiene also required to prevent any of the contamination within the pharmaceutical product. So, thank you so much.